Everyone, today we are going to study about Krebs cycle. So let's start. Krebs cycle is also called tricarboxylic acid cycle or citric acid cycle. It is discovered by H.A. Krebs. Tricarboxylic cycle occurs in mitochondrial matrix. All the enzymes of TCA cycle occur in matrix of mitochondria except succinic dehydrogenase (SDH), which is located on inner membrane of mitochondria. TCA cycle began with citric acid formations. Oxaloacetic acid is an acceptor molecule of acetyl-CoA. In TCA cycle or Krebs cycle, 3 molecules of NADH, 1 molecule of FADH2 and 1 molecule of GTP or ATP are formed in one turn of TCA cycle and 2 decarboxylations occurs. So now let's see the pathway. So in link reactions, pyruvic acid is converted into acetyl-CoA in the presence of pyruvic acid dehydrogenase, in which 1 NADH is formed and 1 decarboxylation occurs. So now in Krebs cycle, oxaloacetic acid acts as an acceptor molecule of acetyl-CoA. So it accepts acetyl-CoA and converts into citric acid, which is of 6 carbon molecule. And oxaloacetic acid is a 4 carbon molecules and acetyl-CoA is 2 carbon molecules. So total 6 carbon molecules. Then citric acid is converted into cis-ecotinic acid in the presence of ecotinase and Fe2+. Then cis-ecotinic acid is converted into isocitric acid in the presence of ecotinase. And isocitric acid is converted into oxalosuccinic acid in the presence of isocitric dehydrogenase enzyme. And in this step, 1 NAD is converted into NADH2. And in the next step, when oxalosuccinic acid is converted into alpha ketoglucanic acid, 1 decarboxylations occur in the presence of decarboxylase. So alpha ketoglucanic acids are converted into succinyl-CoA in the next step, and in this step 1 NADH is converted into NADH2, and here the another decarboxylations occur. The first decarboxylations occur in this step, and the second decarboxylations occur in this step, and here CoH is added. And in the next step, succinyl-CoA is converted into succinic acid in the presence of succinic thiokinase, and in this step 1 GTP molecules, which is coupled with formations of ATP molecule, 1 ATP molecules. And in the next step, succinic acid is converted into furamic acid in the presence of succinic dehydrogenase enzyme, and in this step 1 FADH2 is formed. And in the next steps, furamic acid is converted into malic acid in the presence of furamase enzyme, and then malic acid is converted into oxaloacetic acid in the presence of malate dehydrogenase enzyme, and here also 1 molecule of NADH2 is formed. So basically there is total 3 steps where NADH2 are formed. The first step is this one, and the second step when this one, and the third step when the malic acid is converted into oxaloacetic acid. And in one step FADH2 is formed, and in one step GTP or ATP is formed. So now we know that in one TCA cycle 3 NADH and 1 FADH and 1 GTP or ATP are formed, and 2 decarboxylations occur. Now 2 turns of TCA cycle are required for complete oxidations of 1 molecule of glucose. See here 1 acetyl-CoA is used for 1 cycle. So for glucose there will be 2 acetyl-CoA. So the cycle will be 2 cycle. or 2 turn of TCA cycle are required for complete oxidations of 1 molecules of glucose. Now let's see about the ATP, calculations of ATP. So we know that 1 NADH gives 3 ATP and 1 FADH2 gives 2 ATP. So in one turn of cycle we got 3 NADH2, 1 FADH2 and 1 GTP. So 3 NADH2 gives 9 ATP, 3 into 3, 9 ATP, and 1 FADH2 gives 2 ATP and 1 GTP gives 1 ATP. The total 12 ATP in one turn of TCA cycle. For 1 molecules of glucose, 2 turn of TCA cycle is required. So 2 into 12 ATP. So total ATP in Krebs cycle for 1 molecules of glucose is 24 ATP. Thank you.